Hello, my name is Yoonjin Kwon. I'm in charge of developing analytical method of bioconjugates in video ADC analytical group. Today I'm going to present platform analytical strategies for bioconjugates. I'm going to present in the following order: introduction, analytical capabilities, drug to antibody ratio, free drug related impurities, residual solvents, and conclusion. In this webinar, I will present physicochemical assays specific to bioconjugates, including assays that overlap with antibodies. Now let's get started. The first chapter is introduction. As you know, ADC comprises antibody, linker, and payload. First, in case of antibodies, there are 4 subclasses of IgG, bispecific antibodies, nanobody, and other proteins. Next, there is the linker, which plays the important role in ADC. Linkers can be broadly divided into non-cleavable linker and cleavable linker. Cleavable linkers are in the form of protease-sensitive peptide or acid-label linker. Next, payloads can be broadly categorized into tubulin inhibitor, DNA damaging agents, and topoisomerase 1 inhibitors. For tubulin inhibitors like auristatin MMAE and MMAF, and maytansinoids, including DM1 and DM4. For DNA damaging agents, including camptothecin and duocarmycin. And lastly, there are 3 types of conjugation that are widely used. As you can see on the left side, lysine conjugation occurs randomly and takes on a heterogeneous form. So DAR is distributed randomly, and broad range of DAR. Lysine conjugation have the broad range of DAR. The next one is in the form of interchange cysteine conjugation, which accounts for the largest proportion among commercial products. So DAR exists in the forms of 0, 2, 4, 6, and 8. Recently, there are many cases where DAR is controlled in one form by limiting the conjugation site through site-specific conjugation with engineered cysteine. Samsung Biologics provides a 12-month one-stop service from cell line development to IND filing. We optimize development timeline and simplify project management. In addition, based on 14 years of antibody-based know-how, we have expanded into the bioconjugates field and provide various services as follows. Chapter 2, I'm going to explain analytical capabilities in Samsung Biologics. The analytical capability workflow is as follows: feasibility test, method development, performance test, and documentation. Additional time can be required for bioassay. Today, I'm going to skip the overlap with antibody CQA, and I'm going to focus on bioconjugation analytical methods such as PAR and free drug-related impurities and residual solvents. As you can see this table, most of critical quality attributes are identified by LC methods. The first critical quality attribute is drug to antibody ratio. This is the most important specification for ADC. And also we can assess drug load distribution by anion exchange and ICIF. For purity, we also can analyze size exclusion and CDSDS in non-reducing and reducing conditions. For process-related impurities, free drug, free linker payload, and residual solvents can be evaluated using quantitation method. Also, we monitor the free sulfhydryl quantitation during the in-process control sample. And for characterization tool, we have mass spectrometry for those kinds of methods. AR conjugation site determination and disulfide bond analysis, peptide mapping for post-translational modification, and for antibody oligonucleotide conjugate, we identify the free drug and drug linkers using LCMS. We also have a binding assay and cell-based assay platform in Samsung Biologics. In Chapter 3, I'll explain the drug-to-antibody ratio method. There are 4 types of AR assays. First, Higgs hydrophobic interaction chromatography is the most suitable assay for analyzing native forms of ADC with cysteine conjugation. It requires effort for peak identification. Hence, it is a principle of separation by hydrophobicity. The column screening should be performed considering the functional group of the resin, and then the analysis method should be developed considering the salt type, concentration, and pH for the mobile phase. The second method is the reverse phase chromatography, and this method also can be applied to cysteine conjugation products under reducing conditions. The DAR can be orthogonally confirmed under reducing conditions. Since it is an RP test method, the mobile phase is water and acetonitrile, and the analysis method is developed by optimizing the pore size of the resin and particle size. Also, LC gradient conditions can be optimized. The remaining 2 analytical methods, UV spectroscopy and mass spectro, are also applicable to lysine conjugation. Lysine conjugation is very heterogeneous compound, so those Higgs and RP cannot be applied to lysine conjugation. So for those 2 methods, we can identify the DAR for lysine conjugation. We develop analytical methods in the following order. If there is a platform test method or a customer test method, we perform a feasibility test using this method and then optimize the method for developing under the following conditions. First parameter can be a column screening, considering functional group of resin and particle size and pore size of resin. Next, the mobile phase will be optimized. For Higgs, as I mentioned previously, salt concentration and type pH is the most important. For RP will be water and acetonitrile. Also, sample preparations will be optimized. And also gradient and flow rate injection amount will be optimized based on chromatogram. The next step is performance test will be performed corresponding to the qualification are performed using ICH guideline Q2 R2 items. There must be predetermined acceptance criteria. However, there may be minimal method performance monitoring requirements based on intended application. And after that, internal and customer review, technical report and standard operating procedure and necessary forms are prepared and approved. Characterization using mass can be divided into 3 levels as follows. At the intact level, DAR and post-translational modification can be confirmed. And at the peptide level, conjugation site and disulfide bonds can be confirmed. And for the small molecule level corresponding to the payload, we can qualitatively and quantitatively analyze the payload and payload-related impurities with our QTRAP 6500 model from IX. And for intact level and peptide level, we normally use water synapse access model for large molecules. This is the intact mass data of Kadcyla, one of the products utilizing lysine conjugation. The mass to charge spectrum profiles are shown. The peaks of the glycosylated Kadcyla were observed. Peak of R1, 3, 4, 5, 6, and 8 were observed. And the results are from before the analysis conditions were optimized, so R 3.5 does not clearly appear as the largest peak. But after optimization, the method was determined by gradient condition, deglycosylation condition, and flow rate, and voltage of mass spectrometry, and injection volume. The right table shows the condition of the mass spectrometer. Chapter 4 is free drug-related impurities. For free drug-related impurities, limit of quantitation and limit of detection should be evaluated as they are limited according to ICH guidance Q2. The workflow is on the left side. First, prepare the external standard, and the second step is prepare sample using protein precipitation procedure, and analyze using UPLC with UV detector. And wavelengths can vary depending on the drug because different types of payload will be used depending on the product. So after that, data calculations through software and result will be reported. The right side shows the result of redrug method. As you can see, the upper calibration curve, the R-square value of the calibration curve was also appropriate at over 0.99, and since there was no free drug in the sample, so recovery was confirmed using right sample. And in this step, limit of quantitation and limit of detection will be calculated using several methods. First method is LOQ and LOD can be calculated based on the standard deviation of response of the curve and the slope of the calibration curve. And the second method is both LOQ and LOD can be determined by a signal-to-noise ratio. For LOQ, 10 multiply, and for LOD, 3.3, by multiplying the signal-to-noise ratio. There are 5 important points to consider when analyzing free drug-related impurities. One of them is to determine what substance you want to quantify in terms of specificity. You want to analyze only free drug or free drug and drug linker, or want to monitor drug linker, and quench drug linker. You should determine what to be quantified. And the next step is you better to have a stability data of drug or drug linker, because linker payload normally, this is normally degraded under different storage conditions. So it is quite important to analyze it within half-life. So this data also important for FDRI analysis. The third step is you need to optimize the protein precipitation procedure, considering efficiency of precipitation. Also, thinking about the solubility in organic solvents such as DMA, DMSO, and DMF. And also, we need to consider the batch-to-batch variation for reproducibility. And finally, LOQ and LOD will be determined depending on the calculation, which I previously explained the previous slide. And this chromatogram shows the result of 2DLC, but I'd like to show 3 components: free drug, free linker drug, free quenched linker drug can be analyzed at the same time. This method is developed through the following parameter optimization. The workflow is same as DAR: feasibility test, development, performance test, review, and documentation. For development, we need to consider drug linker instability and protein precipitation procedure, RP column screening, and we should determine wavelength for drug linker. And also, recovery test will be performed using spiked sample. The next method is residual solvents using gas chromatography, flame ionization detection. Organic solvents such as DMSO, DMA, and DMF, dimethylformamide, dimethylacetamide, dimethyl sulfoxide, and isopropanol that are commonly used in the conjugation process. So LOQ and LOD should be evaluated as they are limit tested, similar to FDRI, due to this method is related to the process-related impurities. So the workflow is on the left side. First, stabilize the gas chromatography and FID, and then prepare the standard and sample. We use NMP for internal standard, and each solvents are used for external standard. And also, we use the protein precipitation method for sample preparation. Next, we calculate the data using software and report the result based on the method. Now, our platform method can quantify a total of 4 residual organic solvents, including IPA, DMF, DMA, and DMSO. Solvent with high boiling point is assessed by GCFID. So as you can see on the right side, you can see the GC profile. It shows a good peak profile, and there are no peaks that interfere with the target region. And also, the upper chromatogram is sample solution, and the low one is spiked sample solution chromatogram. So the spiked sample solution chromatogram, you can see the good accuracy confirmed by the spiked sample, including 4 organic solvents. And you can see the internal standard peak at retention time at 13.982. This test method is developed through the following parameter optimization: GC column screening, oven condition, GC parameter, and protein precipitation procedure. The next performance test will be performed by ICH guideline Q2R2 for qualification of analytical procedures used to detect and quantify impurities. And afterwards, review and documentation are performed. For conclusion, there are three major challenges. First, heterogeneous ADCs must be solved through advanced analytical methods. The next challenge is drug. So structure change in camptothecin derivative payloads such as DXD and exatecan can be prevented by pH adjustment. Finally, free payload release and linker cleavage after sample collection can be solved by adding a stabilizer. And also, our payload room has an amber pure light for light-sensitive payload because, in reference, some exatecan derivatives can be degraded under light. So we are controlling under the amber light at Samsung Biologics. So it's also a good solution for those kind of challenges. Samsung Biologics will overcome technical complexity through platform capabilities and leverage comprehensive analytical capabilities and adopt a strategy approach to analytical method development. Thank you for listening.
Okay, great. So our first question is, what are the key challenges associated with the development of the free drug method?
That is a very good question. I would say preventing degradation of drug, which is used as external standard, especially linker payload. Stability of linker payload is also important factor for analyzing free drug and free drug linker. So the next important thing is the proper protein precipitation is also another important factor for analyzing free drug quantitation.
The last question for the webcast is, given the many species of free drug that elute in a profile, can additional free drug species remain unaccounted for outside the window?
During method development and validation, you must evaluate whether you are accounting for or picked. So spiking and recovery experiments can accomplish that. So linkers are highly labeled. So whatever drug species fall off, an antibody will react with something else. So spiking test is necessary for this case.